facilitating today's webinar. You are welcome to join the webinar on the topic Big Data and Artificial Intelligence in Animal Genomics, Opportunity and Challenges, being organized by Guru Angad Dev Veterinary and Animal Sciences University under the aegis of NAHEP component of the undergoing project Institutional Development Plan. Today, I am very pleased to inform you all that we have a fortunate opportunity to listen to Dr. Dinesh Kumar, Principal Scientist, Biotechnology at the Center of Agriculture Bioinformatics, also known as CABIN, at ICRI, Indian Agricultural Statistical Research Institute for the occasion. I cordially invite and welcome Dr. Dinesh Kumar sir among us as the expert of the topic to deliver the webinar lecture. Sir, I extend my heartfelt welcome to all the dear students, respected faculty members, members of the scientific fraternity, and all the other participants attending this webinar. With immense pleasure, I would like to introduce to you to all the person orating today's webinar, Dr. Dinesh Kumar sir.
Thank you. And
Dr. Dinesh Kumar sir has done BSc Honors, MSc Biotechnology and PhD Biotechnology from BHU with GBT National Fellowship and CSIR NET JRF Fellowship. Dr. Kumar has vast works experience of 15 years of molecular genetics at ICR NBGI Karnal. And during his postdoc, he, in the Iowa State University of Science and Technology, AIMS USA, he was the part of the esteemed team which reported the world's first domestic cattle genome sequencing. He has numerous research publications in the reputed peer-reviewed national and international journals like Science and PNAS. And after working in the wet lab for 15 years, he has first biological at supercomputation facility of ICRI SRI. And for the past 9 years, he has been working with his mega team mode in collaboration of multidisciplinary and multiple institutes across the livestock, plant, and fish domains. And he has been in the news for the recent time for developing the world's first crop and animal breed DNA signature server. His team has machine learning techniques in genomics and proteomics data for predictive biology. And he also specializes in the laws pertaining to the IPR and cybersecurities, as he has obtained LLB and LLM. For that, he was a gold medalist in the PG Diploma in Technology Management in Agriculture from the Central University, Hyderabad. I would like to invite Dr. Dinesh Kumar sir to deliver his lecture on today's webinar. If you may, please, sir.
Thank you, Dr. Neeraj. It's great. Thank you. I hope I am audible.
Yes, sir. Yes, you're audible.
Thank you. Thank you, Dr. Neeraj, for this and entire team for this great opportunity to share the experience what our agriculture research team, especially of computational biology in the ISRI, which is the epicenter as on today, in collaboration with our partners, we will be sharing their experience. And let me confess in the very beginning itself that I am not the having any expertise, especially in the area of computational science, but I am being a part or one of the team member, and also as a first biologist of ISRI, maybe I have a better communication skill because I use the biological terms and I understand the process which is happening. So for conceptual and for the applications part, this lecture is well focused, but my colleagues are also online. So if there is any question at the end, definitely our team members will be happy to respond to it. The biological part, definitely I will be trying my best to respond. And computational part, you will have the original developers of the program and the computational expertise of the country. You will be able to interact with them also. So at the outset, let me introduce our team members, Dr. Mir Asif Iqbal, Dr. Sarika, Dr. U.B. Angadi, Dr. Sunil Kumar, and our leader, Dr. Anil Rai, who is also head of the division, and he is ADG ICT, sitting at headquarters, having the dual charge, and he is the main person behind us. And Dr. Iqbal and Dr. Sarika, they are our, I will say, middle cadre scientists, but they are the NAS associate and the Lal Bahadur Shastri Award, NASI Award, all the award you expect at below 40 level they have. And Dr. Rai is our leader. He is the NAS fellow as well. And you can see the small photograph of our students. I will be introducing them at the end of the lecture. So the topic assigned to us is the big data, AI, and genomics, and particularly with the special reference to animal science, because the veterinary university definitely we will do justice if we take example. And in fact, the animal science is the example even for the crop science. Hello.
You're audible.
Yes. So if you see the biological big data, then you will find its feature. In one slide, we just wanted to finish about the introduction of the biological big data. It has 5V, which is the data framework, and it has a 5 process framework, which is 4M. And uh, not always, but most often, the data is voluminous, but not necessarily. Even the simple data, if it is smaller size data, if it is very complex in a non-parametric relationship, then even the smaller data size is also in the definition of the big data. So most often it is voluminous and it has a very high, it has a different variety and has different veracity, like your quality of the data or the different format of the data or the signal noise ratio. So that's also a major challenge, and it has a very high value. And the velocity, especially in animal science, if you see with the reproductive cycles, if you see the disease cycle, then definitely, and if you see the real-time monitoring, especially by the
Yes, sir.
sensors for the efficiency and other things, then velocity do matter, and the real-time data processing is a big challenge, and which we will be able to see. So this is the backdrop of the big data, and if you compare the big data with the YouTube data,
It seems
Facebook
the
data.
slides are not visible.
Hello.
So screen share is not enabled, I think.
Uh, no,
Kindly
I
share your
think there
screen.
was some interruption. So let me restart uh, with the screen share. I am going to share the screen again. Is it now clear?
Yeah,
Yes, sir.
very clear, sir.
Yes. So I guess this was visible to us. Biological
Yes, sir.
data. Yes. So sorry for the interruption. And if we see the genetic resources and big data, then definitely this is going to be very, very voluminous, maybe in next 5 to 10 years. And if you see our genetic resources, maybe of crop, animal, or fish, but let us talk about the animal, then definitely the phenomics data and the genomics data, if you add up, then definitely it is going to be too big. And right from diversity analysis to the QTL, GWAS, genomic selection, or the whole genomes resequencing, all they are going to add big data, and you require some tools, some techniques to analyze them so that the big data becomes the smart data. And that means the information, which is actually the knowledge, if we wish to have the wisdom, making right use of it in terms of the animal breeding or increasing the genetic gain per unit time, then definitely like the precision breeding program, we are dependent on our efficiency to use big data using the AI techniques to make it much more smarter than what it was. And coming to its economic potential, which is very interesting to see. If you see the total global economy, it is around 86 or 87 trillion US dollar. This is the data of 2019. But the impact of AI, which is going to be by 2030, that is just next decade, it is going to be around 16%. It is said that 13 trillion US dollar will be added by putting this AI technique in the global economy. And if you see the Indian economy, where 17% is there and it has a major share, definitely AI has a big impact. If you see even the USDA program, agriculture, AI impact, it's phenomenal. So if you see the sustainable development goal for the SDG 30, definitely AI is going to play a major role. And in that sense also, we must take into consideration that this is not only a national goal, but even the international goal. If you see the velocity by which the data is coming in terms of the years and months, and if you see the data size, if you see the data speed, and if you see the top 500, which is actually technically a supercomputer, otherwise it is high performance computing system, then you can see the genomic data. If you compare with astronomy data and if you compare the sociology data, then definitely by 2025, genomic data is going to be much more bigger than the astronomical data or the sociological data. A standard data set is kept at Harvard Dataverse, which we refer it, especially for big data research work, then it is going to be much bigger. And the moment if you see coming to the genomics part, like what kind of data we are going to have, you can see that your data is the whole genome resequencing. It is going to be the maximum, and your exome sequencing, especially the SRA files, this is going to be maximum because the cost of sequencing has gone down, and they are going to take lead over the protein data or electron microscopy data. So this among the big data, the genomic data is inevitable part, and we have to be able to develop the team to deal with this kind of data in the breeding program. So there is no doubt about it, and these are the factual picture of 2019 with the real data set. So if you see the genomics data, it is increasing, but by 2025, it will be more than the astronomy and sociological data. And the IP traffic will also be increasing, and they will be actually some time limiting if the speed is not appropriate in terms of the data. Maybe if you shift to the cloud mode, then there will be some rescue. Otherwise, it's a big challenge. Coming to the, again to the velocity, if you see the doubling time, it is just 7 months. You can see here the red projected one. So it is going to be in 7 months, the data size will be double. That means in just one day, if you can sequence 3 GB of genome and the cost is going to be so less, then definitely instead of like using the SNP chip, now people will prefer even for the whole genome resequencing, because you are going to have the data in terms of your copy number variants. You are also going to have the data of the novel alleles, which is actually not represented maybe on the SNP array itself, or they are not in enough frequency. So you would not like to miss it. So the resequencing is going to replace the SNP array. There is no doubt about it. Now, coming to the connect with AI techniques, if you see the volume, if you see the variety, if you see the velocity, then definitely to make them further usable, like the smart data, you require AI technique. Without AI technique, this kind of data we cannot handle in the molecular breeding program. And the traditional breeding program has a great limitation. I will say as a layman terms, in the biological terms, that per unit time rate of genetic gain, especially for low heritable traits, that is always limiting. And if you see the gestation period, and if you see the breeding cycle, and if it is longer, unlike the crop, then per unit time genetic gain is actually further reduced. So if we have a technique to put AI into the breeding program, especially by generating the big data of genome and phenome, then definitely rate of genetic gain will be substantially accelerated, which you will see. But taking advantage of these kind of techniques, especially for AI and big data, there are, hello, especially taking advantage of big data, uh, we have to have a different kind of AI approach. And there are three major kind of AI approach. One is your the artificial intelligence itself, then is your machine learning, and then finally the deep learning. You will be able to see the basics because I am not from that field, but what a layman can understand what is artificial intelligence and how it mimics the human brain and how we can make use in terms of its machine learning and also in terms of the deep learning approach. I have some simple example, and this is not new actually. Since 1956, the term is being used by John McCarthy. And now if you see the picture to understand, because repeatedly we will be using the term AI or artificial intelligence, machine learning, neural network, and deep learning. These four terms will be used very repeatedly. So if you wish to understand the basic part, then AI is the bigger word, which actually encompasses all its subdomains. And in AI, you will find machine learning. Within the machine learning, again, there are three types we will see. Then neural network, and finally the deep learning. Deep learning is actually by multiple layer of the neural network, but deep learning can create a data, can understand a data, and can be independent of your operation. Like we tag the photo in our Facebook, or you have a Google Assistant, or you have a predictive writing, maybe when you are writing the email. So these are the example of deep learning. And deep learning can use many things of machine learning, neural network. So these are overlapping actually. And this is the small pictorial representation to understand these terms and their dimensions. So I hope I will be able to move forward with these terms, AI, ML, and neural network and deep learning. Now, again, in the machine learning, which is a type of AI, you will find you have a training data set, you are giving training to the computer, and we call it like supervised. And again, in the machine learning, you have unsupervised also, and you have a reinforcement also. In unsupervised, you are not giving training, but computer is able to train itself. And and we are not giving any feedback. But if you see the third type, reinforcement, where if you see how you rear your child, like when the child is doing something good, your expression is really good, you are happy, maybe you are giving some reward. But when something child is doing which is not acceptable, your reaction is bad. So you are either giving punishment or not smiling, and you are not hugging. So the feedback is there, and trial and error is there, and child is learning. So that is the best example to understand the reinforcement type of machine learning, and which is also used in biological system. Now, when you have supervised machine learning, you have two approaches. One, you can classify disease one and the healthy one. So if you have the data with lot of noise and maybe pertaining to gene one, gene two, and you can classify, and even you can have regression also in terms of survival and with respect to the gene, and you can have a regression line so that your prediction equation starts working. Now, in unsupervised learning, where there is no training data set, if you are using this, then you may have some certain clusters. You can use them in classification, you can use them in association. And here you can see this example of a big Walmart type of store, where the articles which are being purchased, what other articles are getting purchased. And based on these kind of data, many things can be predicted with the season, like what kind of goods will be sold in which season, and this kind of predictive equation starts working. And reinforcement method, which already I had explained, that it is based on your trial error or reward and punishment, the facial expression example I was giving. So this is in short about all three kind of machine learning. Now coming to deep learning, further advancement of it, here we can understand that human brain, and if computer is going to mimic the human brain, then definitely it has to have multiple layer where lot of informations are stored, and then you start using those informations which are actually present based on your own independent logic rather than asking somebody, and then you are making use at various filter level or at various layers. You are taking the decision, like if it is a four vehicle, maybe it's a car, maybe it's a jeep, but you have a backup data, like it looks like a Maruti or it looks like a jeep. So these are the things you can see that you take the logical decisions based on various layers, and finally you land up that yes, you are able to get it. So here in biological system, if you take this example, then in biology we have around 10 billion neurons, and if you see each neuron, again it is by synaptic junction where you have 10,000 neurons are connected, and if you see their transmission, which is based on the voltage or action potential, nerve action potential, your electronic way of transmission, but at the junction there is a chemical way of signal transduction by acetylcholinesterase mechanism. So this way it is working fast. Exactly in case of your deep learning, you have this kind of maybe your different layers or computer network, maybe in this HPC, maybe in CPU, maybe in GPU, or maybe even in cloud. And all are interconnected, and you are able to process the information, and finally you are able to get uh, output, and finally the prediction or the result. Hello, hello, I'm getting some noise. Hello, maybe we can mute the other one.
Dear participants, it is requested that all of you please mute yourself so that we can hear uninterruptedly to the expert lecture. If you may please
Hello,
kindly mute all of you.
hello, thank you, thank you. Hello, I request the admin, maybe Dr. Neeraj, you have the mute the all speakers mic. So please use that, and please allow, please open my mic only. I think you can manage. Maybe some of them are not able to get.
Yes, sir.
Yes, yes, thank you. So you have a simple neural network where you have the input layer, you have a hidden layer, and you have an output layer. Here the hidden layer may be of in multiple layer, and finally output can come. Again, if you see its type, and which is based on the input type of data, there are three different type of your deep learning. One is your convolutional neural network, which we call it CNN, and then recurrent neural network, we will call it like RNN, and then general adversarial network, or the GAN or the GAN. The example is like the image classification, speech, handwriting recognition, or maybe your Google, your auto car driving. And if you see the impact of deep learning, particularly, this is other than machine learning, then definitely there are 19 industries, because this is the McKinsey company, financial company, which claims that it has a potential to add at least 3 or maximum 5 or 6 trillion US dollar per year in 19 industries. That means the data has not only in animal science, but everywhere the data driven decisions has a major role in shaping the economy. Now, if you see this convolutional neural network, and if we see the phenotyping, or maybe your breed identification based on morphometric data, or maybe even the plant identification up to the variety level. Definitely, this CNN based methods can work. And if you wish to see your biotic stress, abiotic stress in timeline, in dose line, then definitely this image processing based on CNN has a great relevance, and this can be used for GWAS study, and IRI is already using this kind of data generation in an automatic system. We have that high throughput phenotyping system, that is the Deshmukh Center, and crop science people, they are able to generate this kind of data. And coming to the breed descriptors of India, like we have your 197 registered breed. And if we put the breed descriptors data in a three-dimensional form, then definitely this kind of approach is going to supplement the DNA-based signature. And how actually it works? It works in a three-dimensional mode. It may work even for two-dimensional, but ideally it is made for three-dimensional image processing, where you have a height, you have a width, and you have a depth. And in case of deep learning, a depth, the color is also going to work. So we have the RBG banding for the color in digital language. So that is also has one advantage. And this way CNN image processing can be done. And particularly for agriculture, the breed descriptors, and for crop, the DUS feature, which is parallel to the breed descriptor, is going to use this kind of data for this kind of predictability. And this is maybe even independent of the DNA. And in case of biotic and abiotic stress, definitely it can work. And if you see the sensor data, especially in the animal breeding, for feed conversion efficiency or the reproductive efficiency or the reproductive behavior observation, then definitely if you have a recording system, then CNN has a major role. And we do not know, like when our animal system for feed conversion efficiency and reproductive efficiency, we are going to use CNN, especially to process the image, and to use your deep learning method to predict what is going on in terms of its impact in timeline or dose line, whatever is there in the veterinary research or animal production. Now, coming to the second type of deep learning, which is the RNN, or the recurrent neural network. Here, the past, it clings into the past. Like when we write in cell phone, few words very often, then cell phone starts remembering, and it has a memory, and it quickly takes that kind of word. We call it like predictive texting. So exactly it is like that. And here, its learning is by its previous input data rather than supervised one. So here the accuracy is better because the input is based on certain the previously approved data. So it starts like the memory, and it has the backup, and it gives edge over the other neural network because you are very sure that for particular this application, this word is good. So this is actually used for language translation and composing of now the music. We can use this kind of approach, and even writing novels with different flavors like Chetan Bhagat, maybe of Shakespeare type, because there are certain patterns which are well versed by the computer, and computer starts using that kind of style, and many new synthesis is happening. This is not only for text, you will even see even for voice, and even for image that you will see in the next type. So if you see the GAN, generative adversarial network, it is very popular. And if you see the your deep learning framework used by Keras is a very popular, but TensorFlow is used by Google, backed by the Google, and PyTorch is actually backed by Facebook, and Microsoft is backing the CNTK, and Amazon is purchased MXNet. And now, if you see its ability, it can take random noise as an input, and it can even generate fake one. and then it can identify the real one. So it has the ability to differentiate between the fake one, but if you wish to make it a negative use, it has a ability to create the original one. And if you have certain image, you want to increase its resolution. If you see the Google map also, so initially you will see by zooming that things are not very clear. But when this kind of GN or the deep learning is used, then image is getting further refined, because it uses that kind of skill to increase the pixel value without distorting the original image, but the initial image was very hazy. So this is in short how we can use deep learning, especially for phenotyping. And deep learning can also be used for genomic data, especially once you have the NGS data and you want to see like the epigenetic modification, then uh, at every data point, you can have the picture that how much methylation is there, epigenetic effect is there, and there are tools for it. And if you want to go for genome assembly, there are certain tools based on machine learning. They are going to help you. If you wish to see the protein structure, deep learning tools are available. If you want to analyze the transcriptomic data, yes, it is there. And ultimately, you will see of our concern is when we use SNP chip and we have GWAS, genome-wide association study, then deep learning is going to play. I will say in terms of molecular marker-assisted breeding, deep learning is going to play a major role. And that I will be covering in only one or two slides at the end, because just that this is for conceptual part, that when we are now doing the knowledge discovery, uh, then at that time, uh, once we generate the genomic data in terms of NGS, then we have six different class of applications, right from epigenetic to the genomic selection of GWAS. These tool techniques can be used. So that also we have to keep into mind. So let us summarize very quickly that big data we saw along its feature and its various process. And we also saw like phenomic, genomic, both are going to generate the data. And we also saw that there are different kind of AI approach, especially the machine learning and deep learning, and their basics, like how we really feel in this world and how they are connected with biological science when we have the dairy farming, and how we can use it in precision dairy breeding to optimize the cost and maximize the production. I will say optimize the production and then maximize the profit. So with that sense, we conclude that AI big data basic part is over. Now let us see in our context and global context with real data set example that how far it is globally successful and what are the constants or will be the constants for our country, and how initiative has been taken by a very humble beginning, not at all, I will say, satisfactory beginning, because a lot of things has to be done, but beginning has been done, so that we will say. Very quickly, we will see how much challenged we are and how much dependent we will be in terms of AI in our agriculture breeding program of all major domain, animal, crop, and fish. So the food grain we have done fairly well, and horticulture 10 times, fish 15 times, milk 9.7 times, egg 48 times. Fine, this is good, self-sufficient, but it is not good enough. The population we are going to have by 2030 or 2050. And this is not good by the time 2090, the temperature will be 4.5 degrees Celsius high. Then that kind of climate change, are we really ready? And if you see the nutritional level of our country, the largest hungry or malnourished population on this planet is in our country. And if you see the diabetic capital, it is our country. So we have many challenges. It's not only the production ability, it's the affordability, it is the biological assimilation ability is also challenged. These are the hint point. Uh, this machine learning and deep learning is going to be used in agriculture research. Now, if you see the pressure we have on Indian agriculture is, we have 17% of world's population, but we do not have equal proportionate claim in water, in land, and our pressure is really very high. And every year we are gifted with one Australia in terms of population increase. And you see, our major problem is population. And if you see the per capita affordability and availability, it is still challenged one. And in post-economic resilience, the pressure on Indian agriculture, because agriculture is the major backbone, it is in pressure, but it has one plus point, that our economic resilience was visible during the lockdown period, and post-lockdown or post-COVID genomic era. We have much more higher hope from the Indian agriculture sector rather than tourism sector or the other export sector, because these are still struggling with a lot of problems. So this is, we are challenged, and AI has to play a major role. And if you see in nutshell, by 2050, everything we have to increase other than population, because that is going to increase inevitably. So the milk requirement, meat requirement, egg requirement, either 1.5 times, 2 times, or 3 times. And we have to decrease our methane production per kilometer square. Otherwise, we are not going to retain this. And we are also challenged, I will say next to COVID challenge, is your rising antimicrobial resistance. And this has reached to the animal science. Even the fish has a very high antibiotic in the food chain. And even the milk has a high antibiotic. Even your animal source meat has the high antibiotic content. And it's going to be a major challenge. It's already a challenge. And if you see the problem of mastitis and the antimicrobial resistance and its loss, it's a huge loss per year. And if you see there is a rise in production level, then your fertility uh, is a major challenge, and around 30% of the elite animals are, what we have heard from our colleague, that they are going to get affected. And we are already in diabetes capital, and diabetes is linked, and many tools are available for high precision phenotyping for low glycemic food. And then we are dependent on this kind of technique for qualitative improvement in the food. And some of you must be knowing, especially for crop science, like Gurmatia, Madhu 55, they are the low glycemic rice, but still we do not know, can we use the GWAS approach to control the glycemic index. But very recently we have a DBT program where one of the trait is glycemic index, and of course we will be using GWAS. Now, in nutshell, if you see like next 20 to 30 years, the progressive or entrepreneurship in dairy will be definitely using AI or ANN or machine learning, right from the drug or medicine to the food feed intake and the entire animal monitoring, including the temperature, everything will be controlled. And the most of the decisions will be based on the data of the animal, and the video recording of the animal, sensor, and all input output will be optimized, and the entire monitoring. Still you can see these things in the world's top milk producing cow, which is in Israel. You will see like it is just like a factory, but it is controlled with so many things that none of the animals are actually present in the natural form. But that is the exaggerated way of my expression. But in reality, the precision dairy farming is going to have lot of big data, and all major decisions of day-to-day animal rearing will be by the big data. And ultimately, the major part, which we can see, the predicting and preventing the disease, that will be the major part. And well-being, definitely that will have a smaller say. And ultimately, ecological practices in terms of methane and other things, they are also going to get controlled. But the major part of machine learning or deep learning will be predicting maybe the genetic merit of the germplasm or the breeding value, what we used to have through BLUP and other technique. So now these data will supplement, and deep learning will be used to predict with the better predictive equations. And this will also prevent lot of disease. So the veterinary cost can be reduced, and that will be the major role. Now, in just one or two slide, if you see how this prediction and prevention can be done. Let us take the example of mastitis. We have now algorithms like BOW or GBT, which is actually based on somatic cell score and the electric conductivity. And you can have a clear prediction that whether mastitis or subclinical mastitis is there or not. Similarly, you can see the other trait based on the different algorithm and different parameters and using the AI technique, and these prediction equation can work. Similarly, the profitability part is if you see feed intake and its efficiency to convert the feed, maybe in terms of beef or meat or egg, or maybe in terms of milk. It all depends on the input data which you are putting in the dairy and the output which you are going to have. So this feed conversion efficiency can also be monitored using various algorithms, and you can have data collection right from your weight, your concentrate of the food, and you can have your body condition score, metabolic rate, and you can feeding weight of dry and your concentrate, and then you can have your RGB camera, you can have RFID for cow feed intake and milk production efficiency and frequency, and you can have a very clear equations using the AI, and the feed conversion efficiency can be enhanced, just like we do it in case of soil science, especially for nutrient management. AI has a huge application. Now, why to use big data with the machine learning and deep learning in animal genomics? Because the previous two slides, those were without using any DNA, without using any genomic data. So now we are coming to the topic actually, that how machine learning and deep learning can be used, especially for your animal genomics data, and for what purpose, what is the motivation behind this. So the very first motivation, which is actually motivation of any classical breeding program or the animal breeder, they have to continue as a leader. So the people understand that biotechnology or computational biology, they are going to replace the animal breeders. Certainly not. They have to be there in our program. They have to lead the program, and these advanced tool and techniques, they are going to supplement, complement, rather than to replace them. So hats off to the breeders, those who have continued their breeding program. And the lot of informations are already generated in last 100 years in the animal breeding program, and that has to be used by these advanced tool. Without those informations, we cannot make best use of these tools. And let me confess, biotechnology, bioinformatics, artificial intelligence stands nowhere if breeding or the classical or the traditional breeding is not there. That must be there, and that program can be supplemented by this, but this alone is nothing, it's zero. Now coming to the health, second major advantage is the health of animal, especially the diagnostics. Diagnostics for production, for your heat, or maybe for pregnancy, diagnostic for the disease, all those things are definitely going to use animal genomics or the genomic data, and we can modulate, we can optimize the production efficiency much better than what it was by the classical breeding, and even the reproduction efficiency can be enhanced. Longevity also, a lot of reports are there. Ultimately, it is linked with your profitability. So the sire selection indices definitely will have certain advantage after getting the breeding value prediction, maybe for the production trait, maybe for disease resistant trait. But if you see the Israeli model or maybe US model where the production has a very high weightage, then you are going to face the same problem which most of the developed country, other than the European country, they have faced. Like high production, but if inevitably you have a problem of disease, ultimately you have to optimize. We will see and their experience and applications of machine learning and deep learning, how to optimize it, you will be able to see. And germplasm management, it's very interesting, especially for Since 2019, our in animal sector also, a gazette notification has been done, especially to identify the animal lines. And two of the animal lines of PDP Hyderabad related to poultry, they have now protection under registration. That means the ownership can be now, instead of community ownership, if any research ownership is there, maybe of private or maybe of government sector, maybe of the farmers, maybe of your breeders, private or public funded, it is going to be protected. So now gradually, of course, animal variety, animal lines, two of them are registered. They are not well protected at par if we compare with the plant variety. But now they are statutorily recognized for the first time in 2019, and we have two lines are recognized. So it is going to be parallel to the inbred lines which we see in crop science. And if anyone wishes to have the protection, even without recognition of the variety, and BPGR, and we have the mechanism of protection of any kind of germ plant if there is some intellectual investment. But in case of animal, that IP protection is still pending, but statutory recognition is there in terms of its identity and ownership. A lot of health molecules they are going to come, and that will be replacing the antibiotics, where these deep learning methods has to be used. And some of them will be from the species itself, and some of them will be across species. And many of them are already identified. Some of them are computationally modified for the better stability, like inducing the cysteine containing amino acids so that it becomes the less temperature sensitive. So a lot of engineered molecules will also be there, and natural origin molecules will there. And if you see the antimicrobial resistance issue, then we cannot replace antibiotics by AMP. Last 2 years publication which we saw, AMP can actually reduce the dose of your antibiotics, and that is also going to help you in managing the antimicrobial resistance issue. Now, this kind of research where the genomic data is too high, it's very costly. And the very first thing, like, should we go for it or not? Can we really afford this? And if this is the investment, when the break even will be happen, like the input cost is compensated by the output cost, or can we lead to some profit? That's a big question. Everyone will ask this before we go for this kind of research. So let us have in one slide the scope and the technology available, which they will generate enough data where deep learning and machine learning can be used maybe in next 20 years or 30 years, even in our country, and definitely within 10 years in the other country. So cattle, buffalo, right from cattle, buffalo to yak and mithun. If you see, the genome assembly is already there. And in case of GWAS, lot of reports you will get, but only few report you will get for camel and yak and mithun. And especially for yak and mithun, you will see bovine HD chip or LD chip has been used rather than their own chip. But reports are there, and taking advantage of their conservation in the genome, reports are available. And even the SNP chip of buffalo and cattle, they are widely used. And in other country, even for your chicken and pig and sheep, it is being used. So now question is, once you have GWAS result, uh, can we use machine learning and deep learning? If yes, then what will be the approach? What will be the limitation? What advantage we can have? Those things we will see. Now, this is the ideal picture before putting this kind of investment, especially for genomics approach for breeding. This is the example of your USDA, and you can see the cows, number of cows is getting reduced. And you can see the inflection point, and you can see here, this is a huge gap, especially 2009-10 onwards. You can see the production is increasing, the profit is increasing, but animal number is decreasing and then going to be constant. So here is the advantage in terms of feed conversion efficiency, in terms of methane production, in terms of your milk production. in terms of optimizing the resources using the genomics tool. But now, even in genomics tool, deep learning has been used in case of cattle, in case of poultry, in case of pig. At least for 3 species, I was able to see some of the references where deep learning has been used. And we will share some of the references that how deep learning is actually helping for which trait. So the moment we take back the previous question, that return on investment, is it really profitable to talk about genomics, bioinformatics, artificial intelligence, deep learning, and we are going to have some return? If that is the question, then the answer is a big yes, but it's a big challenge for us. There is no doubt about it. And if you see the age of parents, which can be successively reduced in terms of selection, because at the earliest, if you are able to predict the genetic merit, your selection starts and your per unit time genetic gain is getting increased. Now, let us talk about the in reality that really this can give some profit, because if your cost of DNA testing and GWAS prediction is going to be higher than the animal per se, then nobody is going to rely on this kind of advancement tool. Because what I will do with the cost of genetic horoscope, if the genetic horoscope cost itself is much higher than the animal per se, so definitely it will be rejected. But if you see the experience of the other country where these things are used, we need not to copy it, but we have to understand the science behind it before we put our money in form of the investment. Then from 2003 onwards, if you see uh, the major change, you can see 2009-10 onwards, use of the chip. And in 2014 data per year, the profit is $84. And if you see the per animal cost in terms of DNA testing, I am very sure it is going to be less than even $10. And if that is the cost of DNA testing, in terms of SNP data generation, and rest is your predictive equation. If you have proper reference population and also you have human resource competence to analyze the data, phenome and genome data, and to improve your equation in your setup, then definitely your predictive equations are going to work better, especially for the larger ruminants, and the return is going to be possible. And here, these are the real example. Now, there are certain limitations in our country, and the topic is also challenges and opportunities. So opportunity, we are able to see how the other world is doing it, but limitations we have to derive from the global situation. So if you see the phenotype data of 2019 publication in Frontiers in Genetics, the phenotype data is huge, more than 10 lakh, and genotype data is really very high, more than 5 lakhs. So you see, this kind of data you won't get even in our EICRP program, be it of cattle, be it of buffalo. So, but of course, this is not mandatory that you need to have this. If you ask a question, that predictive equations can work with the minimum number of animals, you will get many examples where 200 to 400 are good enough to have better predictability than bluff. It depends on the trait with their heritability and the effective population size. So many things are there to affect the equation, but it works even on the smaller data set also. But you have to compromise. Now, if you see the result of SNP chip in terms of your Manhattan plot for a disease like mastitis and foot and mouth disease, you will see sometimes the signal is very clear. Like here in case of mastitis, chromosome 6, you will see lot of SNPs, and chromosome 20, you will see lot of SNPs which are actually associated with this disease. So here signal noise ratio is very clear, and here depending on the correlation with the production trait and the other desirable or non-desirable trait and additive effect, uh, these things, and these things you can put them in the breeding equation or post GWAS analysis or prioritizing the SNP to move forward, you can do easily. But the moment you see this kind of picture, especially for FMD, uh, and its Manhattan plot, a lot of noise you will get. And this is the ideal case where we can have little improvement in our equation or prediction of the breeding value when we use machine learning or deep learning. So we are just trying to give you an idea that once the genotyping result is available and you have a predictive equation, if you are getting this kind of signal, then your selection becomes very easy. And GWAS, post-GWAS analysis to put them in the breeding program for the genomic selection is much easier, but this is much more challenging. But machine learning, deep learning can improve this kind of situation. In reality, when we are increasing one trait, we have to face the challenge of the other trait. And this is the example of most developed country. And here comes your GWAS on genomic selection and deep learning, how you are able to make computational tool using the AI technique for better understanding of the big data to make them really usable without compromising the other desirable trait. Because once you increase one desirable trait, simultaneously your non-desirable trait like disease and other things, disease and fertility, they are always anti-party to the production. That means if we give more weightage to production, then definitely these two are going to challenge us. So the moment we are optimizing, if you see the SAR selection indices, which is of Scandinavian countries, which is very much balanced, they are now giving only 33% weightage to the production and 33% to the other in terms of longevity and disease resistance, so that they can optimize. So American, Israeli models are not going to work. And here deep learning can definitely optimize, and that we will be able to see few examples uh, to make the picture very clear that what actually deep learning does when you are going to analyze your GWAS result. Now, the reproduction efficiency is also a problem, and you can see the conception rate, you can see the carving ease, you can see the problem of stillbirth. This is always there whenever there is production is going up. So production efficiency and reproductive efficiency, they have a very different kind of relationship. We have a lot of QTL data, and this data is available, and a lot of trait association is there. And these data and these equations, if you see the utility of deep learning to understand it at molecular level, these data are very, very important. And once you have your QTL data of past, maybe of around 100 years, these are collected, especially of United States Department of Agriculture. And if you see the transcriptome, and if you see the SNP data, and if you see the QTL data, and if you see the real performance, all these multi-omics data, including your protein types in milk, or maybe MUFA, PUFA, carbon chain length, if it is a beef or meat, all these can be used to understand not only the genetic architect, even to understand the breeding value, and that we will be able to see in next few slide. So this is the result of your 90K SNP chip for 10 years and just less than 500 data, animals, not the data, and 90K SNP chip are related to 6 trait. And if we see here the peak milk yield, and if you see the fat percentage, which is very sensitive in terms of price and in terms of production, and the protein yield and protein percentage, you see a lot of noise in this kind of data. And ideally speaking, the post GWAS analysis requires deep learning to narrow down the pinpoint. I will say that there are two kind of relationship. One is biological relationship, and another is statistical relationship. So in layman's language, if I wish to express myself, then is it casually related SNP with the trait or causally related SNP with the trait? Because LD association always gives a statistical part, not the biological part. And if you have a really good data set and you have deep learning approach to reanalyze or revisit your GWAS experiments, maybe of different labs, maybe of meta-analysis approach, maybe of different location approach if it is of crop science, and even in case of animal science also, maybe of different season, you will be able to narrow down and pinpoint certain SNPs with better noise, better additive effect to put them in the equations so that your breeding value prediction is getting increased. So there is a little difference between animal selection system and crop selection system, but the use of deep learning is to decrease the noise and to narrow down the SNP which are not related biologically, but they are coming statistically. So if we wish to have a better power of discrimination in terms of biological sense, then deep learning is actually required. And that is the message I am trying to pass you as a biologist, that why we need deep learning to understand the GWAS result to improve the predictive equations using deep learning. Now, if you see, you will be surprised, those who are vegetarian and having breakfast, 110 countries, US is exporting egg. And this has genomic selection along with deep learning used. So the product, we are able to feel it, but we sometimes we hardly notice that how much it really impacts in terms of cost, in terms of its your export ability, and the globalization. And if you see the entire agriculture output, this is without even AI. This is much higher than what agriculture output we expect. And of course, this 276, there is a year difference between these two data, but still there is a wide difference in terms of agriculture output by using the genomics program. Now, can we really use GWAS and machine learning and deep learning, how it really improves? Let us see 2 or 3 slides to understand what it really does and what different models are available. And certain models have some plus point, then certain limitations. It all depends on your feature. It all depends on your trait. It all depends on the sample size. And it all depends how accurate you are, how it really works. So lot of variables which is actually present in the mixed model that is going to be further refined using this kind of machine learning approach with the different algorithm. So here we are able to see one example related to blood pressure. I am giving the here we have taken one example of human. If you see machine learning approach, especially for predictive equations and to identify the genes related to blood pressure, then unless you use your deep learning or machine learning, you may not be able to get biologically related genes. You will always land up with statistically associated genes. The message which I was trying to give you, you can see here. And this is the reason post GWAS analysis, once you have your GWAS result, you have to use your machine learning models with various data set. And if it is a complex ensemble model based on your random forest or gradient boosting, and if it is a deep learning, then definitely your neural network. And this can actually improve your accuracy if you are keep on adding your the old data or the previous patient data or the reference data or whatever is actually well validated with the trait data, then your prediction equation will be further improving. In a nutshell, if you see the machine learning models, which is actually used after post GWAS to understand the Manhattan plot SNP and their biological explanation in the cake pathway, that how these structural variants are affecting the epigenetics, how they are affecting the promoter regions, how they are affecting if some of them are changed with the amino acids and some of them are leading to a new kind of mutation. maybe most of them are chain termination. You will see in many of the traits. So if you wish to understand its interaction with other genes and its use in predictive equation, then we have support vector machine, we have logistic regression, or we have decision tree, we have neural network, and every this kind of approach has certain plus point and minus point, which you can see here in one slide. For me, it was very difficult to cover every model. So we thought in our team, we discussed, and finally we thought why to not to put only one slide, because this will be too much. So one slide, we can see the most common models like logistic regression, random forest, gradient boosting, support vector machine, and deep neural network. And you can see the deep neural network. It can handle most of the complex data. That means extremely low heritable traits like mastitis, where the GWAS cannot have a better predictive equation. Then either you have to go by your targeted gene family, gene uh, approach, where you have around maybe 500 or 1000x coverage of resequencing of certain candidate genes, which are actually controlling the disease. And then you have a case control studies for your association, and you have a better predictive equation rather than GWAS result. So ultimately, GWAS result and targeted gene panel both are supplementing each other. But if you wish to further refine and understand and make use in their selection program, then your machine learning is going to help, especially your large complex data with extremely low heritability, and those which are actually associated with very rare allele or maybe a lethal allele or getting eliminated because our selection program is always running. That kind of association you can have by deep neural network. But again, it has certain limitations, like it requires, it has a multiple layers, and it requires your high performance computing system. And it can of course handle noisy data, as you saw in case of FMD, and it can make the right signal. But the easiest method is the logistic regression, which is very easy to implement, and where you have a training data set, and very efficiently you can train and you can start your breeding equations. So we understand that these different models, it all depends the data set, the kind of affordability, the kind of trait we are investigating for. Those things will decide what kind of model you are looking for. Now, for the knowledge discovery part, because till now we covered the applied part, the breeding part, where we were interested to have the better predictive equation for the breeding program. Now for knowledge discovery part, how genes are behaving in a system, and to understand the system biology part, NGS data, once we we have a lot of your deep learning methods available to understand. And NGS data can speak, can talk, can sing well melodiously. If you treat them with your deep learning method to understand that how it really behaves, and this will give you a lot of information to understand about the trait which is happening, and how health is actually uh, acceptable or not acceptable, or animal is getting sick. Those things you can monitor and understand, and if new drugs, new molecules are tried, you can watch them, that how it is actually behaving at gene level, and how it is actually inactivating or how it is activating the entire process you can monitor. So we can see the basic type of the data we expect in a NGS lab. It is the epigenetic modification, then protein and regulatory sequences studies, then splice variants, then SNP in the coding and non-coding region of the genome, which is actually affecting the trait, then protein structure itself, which is also regulatory, and sometimes structural variants also directly comes in the trait, and then biomarker discovery for better production or reproduction efficiency. So if you see deep learning common tools available for methylation studies, SDA and CNN, and commercial name and country is US. Deep methyl, deep chrome, they can be used. And for protein regulatory sequence analysis, CNN and RNN both approach will work. And there are different specific softwares, mostly developed by US, and some of them are by China. And if you see uh, genome-wide prediction of cis regulatory element by your deep learning method, lot of tools are available, mostly developed by Canada. And deep learning for functional genomic data interference, it is by CNN approach. So CNN is taking actually lead, and most of the tools are CNN oriented. And a splice variant also, you will see deep slice made by US of CNN approach. So that if you see the SNP in coding and non-coding, it is by CNN approach. And protein structure and your biomarker, these tools are available. DLN software, which is by the Canada, and US has also developed tool for biomarker using deep learning approach. So lot of tools are available. People are using it for knowledge discovery part. And we just wanted to give you a feel like this AI, ML or DL technique can be used to handle the NGS data or the kind of knowledge discovery we are doing with the specific animal or the trait in question. Now, this was the story part, like how far the world is able to understand the basic knowledge discovery and how the applied work has been done in terms of better predictive equation or better production efficiency, reproduction efficiency, or optimization part, profitability part, those things you have seen now. Now, coming to our part, like in our NARF setup, how this computational center is assisting all other different domains of our national agriculture research setup in educational system, which covers state agriculture university, veterinary university, central agriculture university, and all ICR institute and project directorate, NRCs, and other centers. Now, if we see the very first approach, when we were not realizing in 2013 that how we will be using such kind of approach AI in the days to come. So we just wanted to use some new technique. So at that time, our computational team, especially Dr. Asif, Dr. Sarika, I was having data from the NBSGR, my previous knowledge temple, I will say NBSGR, where we work on GOAT and a lot of more than 50,000 allelic data or fingerprinting data we were having. And we just wanted to set up a computational tool so that automation can be done, and can we have the machine learning approach to decrease the number of locus required, so the cost can be reduced, time can be reduced without compromising the accuracy, and it becomes a model server for the entire world. And fortunately, computational team did really well, and this was developed and it was published, and we felt happy. But at the time, we realized that someday we have to put this into crop system where IP is also protected, unlike the animal part. In case of crop, you have essentially derived varieties also, lot of private varieties also. And if you pirate one variety, there will be some bigger dispute, and lot of scope is there in terms of commercial viability. Take the example of basmati, its taste, and it's a DNA based test and its export value. You will realize that there is a substantial difference per kg in terms of DNA certification if it is certified by the DNA. And we also wanted to develop certain tools where microRNA prediction is much more challenging. Ordinarily, you will see microRNA can easily be predicted from any genome using the existing tool. So we thought that when we are using machine learning, why not to select a genome which is very complex and where the traditional tools are not working, and then our team has the expertise of machine learning. If they select the features based on thermodynamics of your stem loop structure of this microRNA, and if they start predicting, and if we have a validation system that it is predicting something which is not predicted by the earlier software, then it will give a boost. And this is perhaps the longest research paper of our, at least in my career, that it took nearly 2 years, and it was almost rejected, but not written like it is rejected, because a lot of question came, and perhaps 4 or 5 times it was revised, but it was published in Nature group of journal scientific report, and that gave us the confidence that new methodology development how the skill is required and how its validation is much more challenging. And we had been very lucky to have the validation of this kind of tool. And finally, we published this. So the first one was published in Animal Genetics, second one was published in Scientific Report. And then by the time parallel, we had been trying to do this with the protein data also, and we got success in case of fish, when we also got success in case of cattle. So I can say that wherever we got the data, definitely the team did fairly well. If I compare with the rejection of the other papers, here the rejection was almost zero, but the comments and the validation was much more challenging. And lastly, we tried to have our own data set, and we developed one research program partnering Spice Institute of Kerala. And today we have several lead finding, but paper is yet to be accepted. And keeping the requirement in long run, the lesson we had for the breed signature, we wanted to put them in crop signature. And first phase was like you have the normal DNA signature first, and then you try to reduce the number of loci which gives the signature less, so that cost is reduced and accuracy is not compromised, and your machine learning or deep learning has some logic behind it. And in case of GWAS, we are yet to have data from AICRP or any of our animal science institute, but we had been very lucky to have data of GWAS for 300 and more than 300 varieties of wheat from Karnal Institute, IIWBR. Dr. Ratan Tiwari is our leader. And we got data of the live varieties, and their phenotypic descriptors and their recordings, and a few seasons data, a few locations data, and our GWAS result was very successful. And today we have DNA signature for 368 crop varieties. We have DNA based GWAS study using 35K SNP chip of wheat for a good number of traits, at least more than 2 dozen traits we have. And now we have Manhattan plot and other things. But now still we have published these previous information, but let me confess on behalf of the team that we are not having enough data to make an attempt to use your deep learning to improve the Manhattan plot or the post GWAS analysis, because we started only 5 years back. So we need at least 4 to 5 years more to have more data so that we can put our deep learning program. But to have a clear picture in one slide, like how Indian team, especially in agriculture research, is performing, you can see here that we have taken the lead, but we are halfway through. Our first phase is over and we have published. We have very good signals are coming in terms of association and Manhattan plot, but we are yet to have sufficient data to say that yes, Indian team or Indian agricultural scientists are in a position to make best use of the deep learning in terms of the prediction they used to have by the basic bioinformatics tool. So this is the last two bullets are halfway. Now, just in one slide, if you see the animal signature using artificial neural network, which is of animal genetics paper, not very high impact, just moderate 2.8. And you can see here the MLP model, which is giving very high testing performance, 94%. It is able to reduce the number of locus, because the DNA test we had been doing for the that we have published separately, which is not quoted here, by traditional method without using ANN. Normal computational method, we required at least 25 locus to have more than 95% of accuracy. But we used ANN, and then number of locus were reduced to 8 only without compromising the accuracy. And that gave us the confidence that yes, ANN works really well, and it can be used for signature of the breed. And you see, we have not written goat, but the data was of goat, and this was the small humble beginning. And ultimately, we just wanted to see as a biologist that what makes this ANN so successful, especially in this data set. So ultimately, it is the number of alleles present on the microsatellite locus. If it is very high, maybe more than 4, definitely it has to be to have a higher peak value. Then question comes is like you have selected 50 samples or 100 samples, whatever it is, they must be random and your FST must be very high. That means the allelic diversity must be very high, not only in terms of number of alleles present on the locus, multi-locus genotyping, but also your allelic distribution, that is the effective number of alleles. So these three things are actually at the interplay, which our colleague, they have shown you very nicely that how these three things are actually interrelated. So by this kind of a small data set, we were able to understand what actually ANN does. It is doing the prioritization of the locus, which is having more effective number of alleles ubiquitously distributed in the gene pool to become the statistical signature of the breed in question. And it is able to have, based on the features, it is able to have those locus which are not only having high number of alleles, but effective number of alleles is equally high. And their FST value, relative genetic differentiation, which is from 0 to 1. If the two population has same allelic frequency, then FST becomes 0. And if both of them are having private alleles, it becomes 1. So this is also taking care. So I was able to understand and we were able to understand each and every locus, how they are actually contributing. Now, if you see the United States Department of Agriculture, how they have made their breed signature, even for admixtured breed, because in India we have 2/3 non-described or yet to be described population. Those things can also be seen, and that can also be seen by our technique. But when you are putting SNP chip, you have a better position to see whether the animal is pure or mixture of two, mixture of three. Those things are uh, visible. And now this SNP chip based signature we attempted in case of crop because we got the data and we had success. And now we are in our second phase after publishing that variety signature, that how we can use deep learning to reduce the number of locus. In case of wheat, which is 16 GB data, genome is having 16 GB, we have only 54 SNP, from 35,000 SNP to 54. But we wish to have less than 54 if possible, without compromising the accuracy. Then our deep learning or machine learning will be successful, which is only time can tell. In case of your wheat microRNA, you see the genome size and you see the number of microRNA, it's too less. So this was the reason for our motivation, that why not to select wheat, which is most complex, and it is actually hexaploid with mixture of three different genome, A, B, and D. And if you see the age-wise evolution of wheat, A and B are the older and the younger genome is D. So the conservation of the microRNA regions are getting excluded because of the age difference, and this is not going to work. We found one reason with the reference, and then 119 microRNA with the bigger genome. So these are the two reasons, and common reason for every crop or every animal is tempospatial expression. Like tissue-wise, time-wise expression will be different from the zygote to till the senescence or death of the animal or of the crop. So we thought that why not to put a reason that it has less, and we are developing a tool. And till now, the tools available were with the alignment method. Some of them were having other than alignment method also, but most of the tools were based on alignment method. But we thought that this stem loop structure is a thermodynamic property of delta Z, gives the equation if it depends on the type of the nucleotide and its ability to make fold. And if we are able to tap this thermodynamic data, then if 16 GB genome is there, then what are the regions where microRNA can be predicted, or we can have an atlas of microRNA with the prediction ability using this machine learning technique. And if it is really works, and if we have certain predicted microRNA which are not predicted by the previous tool, and if they are really expressed in the RNA library, And if you can really see them with the star sequences, you can also see them in the degradome analysis, where the processed smaller microRNA is present in polyacrylamide gel elution, then sequencing. Then if you have all these proof, then a new methodology has been developed. And this our team did in 2 years time, and it was based on 152 thermodynamic feature of the nucleotide sequence, and the accuracy of earlier tools on wheat was 45%, but we got a substantial jump. In fact, the first comment was of the reviewer was that it's too high claim, but if it is validated, this scientific report encourages this kind of novel finding, which will be relevant for the other polyploid crop like tetraploid crop, is the cotton and other things. So we also got excited, but the problem was how to validate them. But fortunately, we got RNA library in the NCBI. We also got the degradome, and fortunately, our predicted one were present. And in 4 or 5 revisions, finally, this paper got accepted and gave us the confidence. Then for antimicrobial peptide prediction, we had the motivation of antibiotics resistance and its residual contamination, then your export rejection. And if you see the Nissin, which is protected by patent, and it is your discovered by computational method, and 4 or 5 others are at clinical trial. And if you see the veterinary cost, and if antimicrobial peptide, if we can discover by taking advantage of the cattle genome itself, by assuming 30,000 genes, some of them are antimicrobial or all, let us prove it computationally, because there is no cost involved into it. And finally, we did, and we had the success using machine learning. And if you see the aquaculture loss because of this biotic stress where antibiotic is used, it's huge. So if $50 billion is lost, and if you have antimicrobial peptide prediction system for the aquaculture fish specific, this is definitely going to be used and it will be popular. So we attempted one on fish, and that was also successful, and we published. So now the global market is huge for antimicrobial peptide, especially looking at the antimicrobial resistance problem. And if you see all third world countries, a very interesting paper came, of course, after publication, which gave us the confidence to look to continue this kind of work, that all third world countries where the moisture, or the tropical countries where the humidity or the biotic stress is very high because of the conducive environment, antibiotics use is also high, and the resistance is also coming to the animal, affecting the human health also. So this is the uh, directly from the paper of science, just to convince you that yes, it is no longer the animal sectors are away, antimicrobial resistance is affecting our those domestic animal also. Now, this antibiotic has a risk for human, especially for colorectal cancer. So this research, it's not that it is related to human, it is related to our domestic animal and domestic animal product. Coming to the use of antibiotics, if you see, especially Economic Times report, especially hub of Hyderabad, which is hub of the antibiotics, lot of resistance report on random sample has been taken, and that this has been in the news. So we continued our AMP prediction using machine learning approach. And we had 2 paper, one for cattle and one for fish. Now, time is less, so I will be moving little faster. This is the last approach using computational tool, especially your artificial intelligence for their prediction. And in case of cattle, we assume that all of them or none of them are actually antimicrobial peptide. So we just took the known antimicrobial peptide specifically of cattle, and that we tried to go by species specific approach, because we thought that cattle is a warm-blooded animal, fish is not the warm-blooded animal, and a protein structure is dependent on the temperature also. So why not to make best use of this your species specific approach, and let us see can they predict. And if all known wet lab validated are again running well in our system, in our software, which is developed, then we can believe that yes, sensitivity, accuracy is well, it is not compromised. So that kind of validation was also done just to prove it, because that is also required if you are publishing it with known, you have, your tool must respond to it. So that was done, and finally we had the success. So when the features were selected, these are the common features you expect in the any amino acid and its particular sequence, like your composition, which is of 20 type, then molecular weight, isoelectric point, and then it's all half-life and aliphatic, water loving, water hitting, all those things comes into the picture because they decide their stability. And we used ANN and SVM, both the techniques, and finally we found that support vector machine methodology is much superior to the ANN, and that was implemented in our web server, so that you can put any gene sequence or amino acid sequence or protein sequence, and you can ask the server that can you tell this is the antimicrobial peptide or not. What advantage I will have as a biologist? I need not to go for bioassay of zone of inhibition or minimum concentration of inhibition using the standard antibiotics. We can directly first do the in silico analysis, then we have targeted molecule. Those we can go for bioassay. So this will save time, save cost, and that is the purpose of any bioinformatics tool development. So this is the screenshot of our cattle server, which is actually working for AMP prediction, and you have to paste. You can upload, you can have your test sequence, which is already available there, and any one of you can access this. And once you click, it will tell whether along with the SVM score, it will predict whether it is antimicrobial or it is not. And then you are sure to go for its validation or not. This is for your fish exactly same way. You have to paste the test sequence, then it will predict whether it is your antimicrobial or not. This information you will get. And then we thought, why not to use spice as a plant by challenging with certain biotic stress and then expecting that it is going to give certain specific antimicrobial peptide. And that we can see by proteomics data, and we can also see by transcriptome data, because till now our experience was with the genome data after the cattle. So we generated transcriptome data, and based on that we found certain lead molecules having antimicrobial activity, and we also saw the pattern for their stability in terms of methionine, because these are actually used in the downstream processing when you are going for industrious mode of production, especially for AMP at higher scale. So that kind of screening, these are the unpublished results, but we got lead finding in this, and you can see their sequence, and you can see the antimicrobial predicted probability, they are present, and some of them are validated at very small scale, but frankly speaking, the actually required validation is still pending. We are looking forward for it, but we are running slightly slow for this. And this is based on proteomics data after challenging the plant, and we got antimicrobial peptide molecule even at peptide level, and these are the details of uh, lead finding. So in summary, we have 97 antimicrobial peptide with very high cysteine rich motif, which can be thermostable based on transcriptome data. And we have 24, at least minimum, based on proteomic data. We have lead molecules, but they are yet to be validated at larger scale. And in validation, we expect that they are from the spice, but they can be validated in terms of applications in crop. They can also be validated on fish, because you know the spices are put for antimicrobial nature, like your pickles and other things. So they have a wider applicability across domain, and bioassay is pending. And if we quickly summarize, these are the three publications of our team on antimicrobial peptide. Coming to the signature part, only one slide. Now maybe two or three, the lecture will be over. So we have a web server where we wanted to have signature using 35,000 SNP, but minimized to 54 only. It is just like having a Aadhaar card for every individual. Instead of 16, you have 54 alphabets or digit. And still 54 can be minimized if we are using the deep learning. So we have reached up to 54 without putting machine learning or deep learning, but further reduction is possible, and that we are yet to have, but we are on track for it, and we will go for it. And here someday we will have successful story using deep learning. But computationally we have 54 SNP of 16 GB, which has a signature for every variety of wheat actually present in country as on today. And these 54 are with the QR code. So if you have a Sarbati type of wheat, especially from MP, and just like Basmati, you want to have the scanning of the product to have consumer protection, then 54 SNPs will be there. And if there is any deceptive indication, or there is a cheating, or if this gets a GI status, and if GI protection is required for benefit to the producer and also to the consumer, those things it will take care. And we have given example of IP dispute for biscuit making ability in this paper for wheat, which was actually settled, and that was a bigger bigger dispute, and as your DNA approach was able to resolve this, just like your Basmati and non-Basmati. And we have developed, our team has developed, especially Dr. UB Angadi, who is our computer scientist, perhaps the best one ever we have seen. And he developed the tool. This is in-house tool, zero outsourcing, and this works well in Android. And this is the world's first tool which can analyze the DNA signature of any crop variety, because this was our model crop developed by SRI IWBR in a joint project, and it starts predicting. And this is the first handheld which is taking care of your SNP chip data as well. And when we went for GWAS with the different trait, we have two publications. You can see these two publications are very sensational publication, but in case of animal, we confess that we are yet to have. And recently our paper was listed in top 3 paper of the world, especially for Rust. And this was listed on Borlaug Global Rust Initiative, BGRI newsletter, and also at Cornell University. And you can see our 2 paper. One is related with the morphological trait, where we have the Manhattan plot, we have our association, but we are yet to use deep learning to further dissect the problem. And in case of 3 biotic stress, like your stripe, stem, and leaf rust, we have these 3 were challenged, and we have the GWAS analysis, and this was the very hot paper. And you can see its impact across world within 100 days for both the publications. And US and China, you can see here how much people are accessing. And this is the global map, the impact of the paper. And this is the second paper as per the rust resistance. And you can see the readers are more in US than even in India or in China. And this happened after post-COVID. This is the June 2020 publication, and within 100 days, our publications were very hot. And I am sure that the day will come when our team will be using deep learning, and we will improve the method, just like software. India has to be leader in computational. That is the dream we have. And we worked with the your CIRB and NDRI with Dr. Datta and Dr. Waris. And we also made an attempt for candidate genes, but here we are yet to put the machine learning. But this paper is under revision. But if larger data comes, definitely we are moving forward. But this is very much in our mind. And major challenge for Indian R setup is the phenotype gap. You saw like how the other countries, they have phenotype. We do not have. Even in AICRP also, if you feel like every animal is having phenome and genomic data, uh, of course, some of them have, but not all. We have to accept. And high precision phenotyping, if it is not economically relevant, ERT we have written here. But unless we have precision phenotyping, we cannot have advantage in terms of feed conversion efficiency. And if you see the somatic cell scoring, it is done at limited center. But it's not that somatic cell score based Shire selection indices is actually used everywhere in the breeding program. So that part we have to accelerate, and multi-omics way of integrating, like the proteomics, transcriptomics, epigenetics data, all putting the machine learning to understand and to have a better predictive equation. Those things are really pending. And masters and PhD program, the time has come that optional course must be there for AI, even for LPM, even for your genetics, even for biochemistry, even for physiology, because this biological science is going to be the computational science. If IIT has made compulsory biology in BTech first year, 5 years back, that means multidisciplinary research has to be there, and biology and mathematics, if they are not together, I am sure it will be difficult. And this is the word of the first biologist. Like I am the person sitting here is the least aware person about my own competence, because I am the least competent person. But I am most aware of the value of statistics very precisely, be it bioinformatics, be it statistics, be it your artificial intelligence, including the deep learning. We have to change our mindset. And I'm very happy that Veterinary University of Ludhiana, GADVASU, has taken the lead, and I appreciate the entire patron and the dean and the head, including Dr. Neeraj, for selecting this topic to sensitize the students that how much challenging it is, and we have to make best use of it. Otherwise, we are putting money, we are rearing the animal, but if we can have more profit by this kind of tools and techniques in long run, we have to have this. And let me confess, there is a issue of policy blueprint. If we compare USDA, if we see INRA, if we see Zyro, they have the priority of the trait, priority of the species, and then money comes, more money goes for phenotyping, phenome. But in our country, more money goes for genomics, more money goes for biotechnology, and there is a hype. Biotechnology will not talk with the breeders, and people will feel breeding is a dead subject. So this kind of policy mindset, we have. This is also a major challenge. We have to now reorient ourselves, and ultimately more R&D is required, because sitting in tropical country with the huge diversity in the animal breeds, uh, we understand that biological pressure is very high, and if the pressure is very high, there is a need to understand that the variants statistically, biologically, their correlation with large set of data. So in fact, we need much more of the deep learning tools and techniques to bear our own challenges, whether it is abiotic stress or biotic stress, especially even in animal sector. This we have to understand that our requirement is very high, and post GWAS analysis prioritization is a critical part. And most of the time we understand that if GWAS is done, when paper is published, some tools are recommended or some SNPs are recommended. Most of our breeding program in our country, we never feel like post GWAS analysis, we have to do it. Maybe in next 5 years, maybe even in 10 years, but nobody is talking about this. And nobody thinks that how the investment of this high-tech research is going to be used in the breeding program to increase the profitability per animal, as we have seen in the previous slide. That kind of approach is actually required to supplement the traditional breeding program. But most of the time we feel traditional breeding program itself is likely to be endangered because these advanced tools are there and our hype of biotechnologists is there. So the time has come that breeders has to take the lead, be at the first line and be the leader. Let us accept this. And biotechnologists, computational biologists, all bigger hype or your high-tech weapons, they have to take a back seat. And if required for the trait, especially for low heritable trait, they have to be in the business with right kind of data set, right kind of equations, right kind of competence, rather than disturbing the traditional breeding program. So these are the challenges we have just tried to enlist rather than getting it copied from somewhere. So this is highly localized, Indianized, and let us take it in true spirit rather than something like the criticism or yellow journalism. that I'm talking too negative. No, these are the point we have to take very scientifically to make best use of the talk in terms of long-term planning, and also in terms of teaching, and also in terms of enriching ourselves. And lastly, these are the team's contribution and publication. You can see the first part in 2020 shown in different color, and this is just last 5 years output. It is more than 30, and we have huge funding even in post-COVID. And it starts with the Dr. Datta's project of NDRI, Bill Melinda Gate, CIRB is partner, Bangalore Center of NDRI is also our partner, and CIRB, Dr. Waris is also our leader from that center, and our team is also involved. And we have DBT funding of a mega project, wheat, rice, sesame, and Dr. Sarika is PI of rice of more than 50 crores. And of course, our center has less money, but the cost of data we understand it's huge. And wheat, our center is also there, and the cost of wheat and sesame is 38 and 39 crores. And we have Center for Excellence for Bioinformatics of more than 10 crores by DBT. These are all post-COVID. And we have NSF project with Dr. Datta. We have Gujarat Biotech project. We have DBT Twinning program, which Dr. Asif is the PI. And we have uh, Mango Genome project or Functional Genomics project. This is the earlier name NPTC. Mango Genome work has been done at this center. Any day we will have a bigger publication. And of course, we have a network project of ISRI where 16 different partners are there, where the CIRB is one of them, then CIRC, CIRG, GOAT is also there, but other centers are not there. Of course, IVRI is there, but FIS CFA Mumbai is missing. And the uniqueness of our team is multidisciplinary and mostly led by the breeders, not led by the biotechnologists. That's the unique thing. And we believe in speed, we believe in collaboration, and we believe in knowledge partnership in the era of your knowledge economy rather than pastoral or industrial economy. And I'm extremely thankful to our students. They are better than us. There is no doubt about it, and that is the way we have to go for intellectual breeding. And uh, if you see Vasu, see, he is having more than dozen paper during PhD, and I'm sure much better than any scientist and any average scientist of our system. And many of them, they have completed PhD, and some of them after 2 years publishing some paper, they have gone abroad for PhD. They are getting good fellowship, and we feel happy that we have migratory bird and they are doing so well. And if parent line and the progeny, if the breeding value is less, parental line has to be discarded. But this we hardly practice, but we firmly believe in intellectual breeding, that in long run every student has to be better than us, then only our country is going to have benefit. But if we compete or if we are better than our student, I think it's a dead end of breeding, be it animal, crop, or intellectual or scientific breeding. So we firmly believe in this. We learned this from our breeder, and we acknowledge, especially to our Honorable Vice Chancellor, Dr. Indrajit Singh sir, who was actually director of our Buffalo Institute, where our keeping program was running. He was very supportive to us. And we are also thankful to the Dean, College of Veterinary Science, Dr. SPS Guman sir. And we are thankful to organizing team, Dr. Neeraj, and co-organizing secretary, uh, Dr. Samarjit Kaur, Madam, and Dr. Bharti Desmukh. And we are also thankful to the entire team and the students and the delegates, those who have participated. And on behalf of the entire team of NAHEP, especially GADVASU Ludhiana, we are thankful to ICR, especially to SMD, Education Division. Our institute also belongs to the Education Division, especially for the NAHEP support in form of a project, because in this way I am sure your center in Ludhiana, though it's a new university, but doing really well. And I'm proud of some of your faculties whom I know, And from their profile, I guess the university is the perhaps the most happening veterinary university ever I have seen. The traditionally well-known bigger universities are there, but I have a special place in my heart just because I have seen the beginning of this university and how things are moving. And thank you so much to everyone. And I am sorry if we spoke something which is not that scientific, may not be that palatable, but please excuse us in true scientific spirit. Thank you so much. And this is the time to learn from your questions further. And my email ID is very simple: Dinesh Kumar, BHU, Banaras Hindu University, without any dot, Dinesh Kumar BHU at gmail.com. We look forward for all interaction and we will try to respond. Our team is also here. So thank you so much once again to everyone. Over to you, Dr. Neeraj.
Thank you very much, sir. Gentle reminder to all the participants that a link has been shared on the chat box for your registration. That will be available even after 10 minutes of the this session. So you kindly register to get a certificate, e-copy of the certificate. Uh, and after that, coming to the last section of the today's event, on behalf of all the attendees and the uh, university, I take this opportunity to express my deepest gratitude to the speakers of today's webinar and the leading figure in biocomputation in ICR system, Dr. Dinesh Kumar sir, and his team for sparing their valuable time and sharing the vast knowledge and experience they have created in the field of genomics and biocomputation, with a special emphasis on the artificial intelligence and machine learning. Thank you, sir. Thank you very much for your lecture. Thank you very much.
Thank you.
I would also like to express my sincere gratitude towards our worthy Vice Chancellor, sir, Dr. Inderjeet, sir, Dean COVS, Dr. Ghuman, sir, all the authorities of the Guru Angad Dev Veterinary and Animal Sciences University, Ludhiana, and the Indian Council of Animal Research for providing this golden opportunity to the Department of Animal Genetics and Breeding to organize the event, besides providing all the necessary facilities. I also thank all the members of the IDP team and faculty of the Department of Animal Genetics and Breeding for their active participation and support for the webinar. Last but not the least, we extend our heartiest thanks to all the participants for their immense interest and patient listening to the webinar. Hope all of us have learned a lot and enjoyed a lot this webinar. Thank you. Thanks a lot.
Thank you.
Thank you. So with this, this webinar, we think that webinar is concluding with this. So I would at last thank each and everyone who have contributed, attended, given suggestions, and in any way contributed to this webinar. Thank you, one and all.
Thank you.
Thank you, sir.
Thank you. Thank you, everyone.